Welcome back. In this video, we are going to continue our discussion of adaptive immunity by looking at the structure now of the T cell receptor, including its interaction with MHC molecules, which are important for the process of antigen presentation. So let's remind ourselves of some of the molecules involved in the activation of T cells. You may recall that there are two major types of T cells that we've covered thus far in the course. One of those was cytotoxic T cells, or T subscript C, as you can see here. And these cytotoxic T cells, like all T cells, have a T cell receptor, or TCR. And the TCR can bind to antigen, but unlike the B cell receptor, requires something called antigen presentation, where an additional molecule that is displayed on the surface of the neighboring cell, in this case an infected cell, the antigen has to be presented by a molecule called MHC class 1. And MHC class 1 uniquely presents antigen to cytotoxic T cells. And cytotoxic T cells, for this interaction to occur, they use an additional molecule called CD8, which serves as a co-receptor to the CDR. And it's the tripartite, or the interaction of all three of these components, both TCR with MHC class 1, along with the CD8 co-receptor. All of them are required for successful docking and presentation of the antigen to the TCR and the transduction of signaling downstream within the cytotoxic T cell. And when the cytotoxic T cell becomes activated, its main effector function is going to be cytotoxicity. It's going to kill usually the cell that's presenting the antigen, in this case the infected cell on the other side. In addition to cytotoxic T cells, we also saw that there were T helper cells, or T subscript H. And instead of CD8, T helper cells express CD4. This allows them to interact with antigen being presented on MHC class 2, which was uniquely expressed on the surface of cells we call professional antigen-presenting cells. These were cells like monocytes, macrophages, dendritic cells, and B cells. These cells are typically not directly infected, but are taking antigen in from the outside through endocytosis or phagocytosis and displaying those antigens on MHC class 2. And again, the interaction of all of these components, MHC class 2 with CD4 with the TCR, is required for the activation of the T helper cell. And when it becomes activated, its main effector function is going to be the production of cytokine. So I just want to put all of these molecules back in our minds, and let's sort of look at each of them in terms of their structure and how that structure underpins their ability to interact with each other. So if we start with the TCR itself, and the TCRs are identical between CD4 and CD8 T cells. They have an overall the same structure, even if the antigens that they bind to are different. In terms of what is in common, we see that most conventional T cells have their TCRs have two chains, an alpha chain and a beta chain. There are such a thing as gamma delta T cells as well, but that will not be the subject of our video today. By far, where we should always begin when we're learning about T cells are the most normal, usual types of T cells, which have TCRs made up of an alpha and a beta chain. And we can see that the structure of the TCR maybe looks familiar to you because it reminds us of the Fab domain of the immunoglobulin. Just like the Fab domain, there are also a variable region and a constant region of the TCR. And so then that gives us a variable alpha, a constant alpha, variable beta, and a constant beta. And again, the variable region is going to be towards the N-terminus of the TCR, and the N-terminus is where antigen binding is going to occur. Similarly, the C-terminus corresponds to the constant region. It's going to be closer to the membrane, and it's going to be more involved kind of in structural integrity as opposed to antigen binding. And again, if you compare the TCR of one T cell to the TCR of another T cell, where the most variability in terms of the sequence of each individual TCR is going to be found are going to be in the most N-terminal residues of the variable region of both the alpha and the beta chain. And you can see that when you look at structural diagrams of the TCR, like you see here from this paper that I'm referencing here at the bottom, they have a very nice illustration of when we look at the T cell receptors shown here, and we can see these different segments that we saw on the last slide. If we look at a diagram that sort of shows the way that this protein folds on itself, you can see that the residues, again, that are at the most N-terminal portions of both the alpha and the beta chain, we refer to them as CDRs, or complementarity determining regions. Again, as we saw with the BCR or with immunoglobulin, another name for CDR is hypervariable region. So you can see that both the alpha and the beta chain have 3 hypervariable or complementarity determining regions. And these regions, again, are going to be at the most N-terminal tips that are going to be the most proximal, not only to antigen, but also, in this case, to MHC. So the CDRs need to come together to make a unique shape that recognizes the complex of MHC and antigen, because it is the combined shape of antigen as it's being presented on MHC, which really serves as the ligand for this receptor. Just like immunoglobulins, we see that there are hypervariable N-terminal sequences known as complementarity determining regions. So I'm stressing that the TCR interacts not only with antigen, but also with MHC. So let's look a bit at the structure of MHC. If we start with MHC class 1, you can see that it's a little bit of an unusual shape. It is made up primarily of an alpha chain, which has 3 subunits, alpha 1, alpha 2, and alpha 3. And the alpha 1 and the alpha 2 subunits, when they fold, are actually going to be what forms the peptide binding cleft, meaning the sort of pocket that forms on the molecule that the antigen can sort of sit inside of. And if you look at a sort of a top-down view of the MHC class 1 molecule, you can see that the alpha 1 and the alpha 2 subunits, the way they fold together, forms this neat little pocket. There's kind of a helix structure from each subunit on either side. And this is where the amino acid the sequence, that the short peptide that we use as the antigen that we're presenting, it's going to sit sort of very nicely inside this little basket that's being made here by these 2 subunits. In contrast, the alpha 3 subunit is more structural, and in addition to this alpha chain, MHC class 1 also has an additional subunit known as beta 2 microglobulin, and this basically serves as something like a chaperone, or sort of a structural integrity, something that serves the structural integrity of MHC class 1. It keeps the alpha chain folded and stable in this particular conformation, so it's an important part of MHC class 1, although beta 2 microglobulin is not involved directly in holding antigen and presenting it, which is in this case served by the alpha 1 and alpha 2 subunits. MHC class 2 is similar in many ways, although it is made up of a heterodimer of alpha and beta chains, and you can see that there's an alpha 1 and an alpha 2, as well as a beta 1 and a beta 2. And for MHC class 2, it's the alpha 1 and beta 2 subunits that form the peptide binding cleft. And you can see a similar kind of structure emerges where you have these 2 helices, one from each subunit, and the peptide sort of fits again nicely in between the 2, and that's where it's going to sit and be presented. So for MHC class 2, it's alpha 1 and beta 1 that make up the peptide binding cleft, whereas alpha 2 and beta 2 are, again, more similar to the constant regions of antigen receptors. They serve more structural purposes. Now that we've seen the structure of both MHC class 1 and MHC class 2, we can think about the way that they then interact with the T cell co-receptors. So starting with CD8, which again is going to be expressed on the surface of a cytotoxic T cell, which we often just refer to as a CD8 T cell. One of the reasons we need CD8, or why it's an important part of that interaction, is because CD8 actually interacts directly with MHC class 1. And both the alpha and the beta, so I should say that CD8 is also a heterodimer of alpha and beta chains, and both of these chains physically interact with MHC class 1 primarily on the alpha 3 subunit. And so even though alpha 3 again is not used to bind directly to antigen, we do use it to associate with CD8. And that's going to be an important interaction ultimately that sort of holds the entire complex of co-receptor, TCR, and MHC, in this case MHC class 1, together. So this slide is showing you the structure of CD8 as well as how it interacts with MHC class 1. CD4 looks a little bit different compared to some of these other molecules we've looked at. It actually has 4 immunoglobulin-like domains but it's actually the D1 domain that interacts with MHC class 2, and especially, again, the sort of beta 2 subunit of MHC class 2. Um, and this interaction also, again, is required for all of the components of the TCR complex to fully dock together and transmit a signal. So here on this slide, you can see that the structure of CD4 is just a little bit unique, a little bit different compared to some of the other mostly kind of dimeric proteins that we've been looking at. But also is essential, again, for the antigen presentation to CD4 T helper cells. In addition to MHC, either MHC class 1 or 2, or the co-receptor CD4 and CD8, of course, we have the T cell receptor itself, and the alpha beta chains of the TCR are what are important for antigen recognition, but there are additional elements of the TCR, including, or I should say of the TCR complex, which include not only the alpha beta chains of the TCR proper, but an additional molecule known as CD3, which we can also think of as a co-receptor. And the CD3 co-receptor is a bit more integrated with the TCR itself. And so you can think of an overall TCR complex, which kind of has two parts or two modules, if you will. There's an antigen recognition module, which binds directly to antigen, and that is made up of the alpha beta chains of the TCR. And then there's sort of a signaling module, which is made up of the CD3 co-receptor. And the CD3 co-receptor is a bit of a complex structure. It contains three different dimers. There's a heterodimer of what we refer to as epsilon and delta chains. There's another heterodimer of gamma and epsilon chains. And then finally, there is a homodimer of zeta chains, as they are called. This is the Greek symbol for zeta. Notice that these molecules have cytoplasmic tails that extend deeper or sort of further into the cytoplasm, especially the zeta chains. And their positioning in the cytoplasm is actually what allows the overall TCR complex to actually transduce intracellular signaling. The TCR itself, the alpha and the beta chains, they do have cytoplasmic tails, but they themselves have no intrinsic ability to signal. They're very short, and their main function is simply to anchor the TCR into the membrane. We need these various chains of the CD3 co-receptor in order to actually interact with various types of enzymes and other signaling molecules in the cytoplasm for any kind of intracellular signaling to occur. So if we kind of put all of these pieces together, we get an overall structure that we might think of as the immunological synapse. And the immunological synapse gets its name from the similarity that it holds to a neurological synapse between two neurons. You can see that two cells come into close juxtaposition. In this case, we're using antigen presentation via MHC class 1 as an example. And so on one side, we have an infected cell, which is presenting some kind of antigen on MHC class 1. And in this small space between the infected cell and the CD8 T cell, there's a platform of many molecules that are facilitating the transmission of information from one cell to the next, which is what makes it a synapse. In the immunological synapse, that's conveying the information that in this example that we have infection in the presenting cell. It's presenting via MHC class 1. That is docking to the TCR of the CD8 T cell with the help of CD8. And now kind of notice that when we add all of these other components, that CD8 actually it actually even bends over to be able to interact with the MHC class 1 molecule. Notice that all of these physical interactions are important. But we also have now, which we haven't been picturing before, the addition of the CD3 co-receptor complex. And these cytoplasmic tails of the CD3 co-receptor complex, when all of these pieces come together, will actually facilitate signal transduction within the T cell itself in order to facilitate the things that need to happen inside of the T cell. Because this is a CD8 T cell that's going to induce signaling properties that are going to result in cytotoxicity, the transmission of information back to the infected cell that is going to induce apoptosis. But we could draw a very similar picture with a CD4 T cell, in which case we would have a professional antigen presenting cell presenting its antigen via MHC class 2. We would see CD4 here instead of CD8, and then we would see that CD3 would be transmitting signal transduction leading to the production of cytokines within the CD4 T cell. And an additional term that I want to introduce here is that this constitutes something that we refer to as a ternary complex. Ternary giving the idea of three things, three elements being involved. I also used the word tripartite earlier, which has a similar definition. The ternary complex of these three key molecules in MHC, either MHC 1 or 2, the co-receptor, either CD4 and CD8, as well as the overall TCR complex, the constituents of which are TCR alpha beta along with the CD3 co-receptor. This ternary complex sort of forms the nucleus or the main part of something we refer to as an immunological synapse. There's some other elements of the immunological synapse which are not shown here. This is actually what is sort of the most important for inducing signaling and effector function within the T cell itself. Okay, so summarizing our discussion of the structure of the TCR and MHC, we saw that TCRs are also immunoglobulin-like molecules that can recognize antigens, and they are made up of alpha and beta chains, which are sort of together reminiscent of the Fab domain of an immunoglobulin molecule. And we saw that these alpha and beta chains are also associated with the molecule called CD3, which contains multiple chains, including deep intracellular zeta chains, which are important for intracellular signaling. We saw that TCRs also require co-receptors for full activation. In cytotoxic T cells, TC, that is CD8, which interacts with MHC class 1, whereas in T helper cells, the co-receptor is going to be CD4, which interacts with MHC class 2. Finally, we saw that a full ternary complex is required to initiate TCR signal transduction. That ternary complex included the TCR, the overall TCR complex itself, which includes the TCR alpha beta chains along with CD3, as well as the co-receptor complexes of CD4 and 8 and MHC. And all of these molecules need to bind together to get a full event of antigen presentation and effector outcomes within the T cell that's receiving or binding to whatever antigen it is that's being presented. Okay, so that covers it for this video focused on structure. In our next video, we'll actually talk about more of the molecular signaling involved when you successfully put this ternary complex together, signal transduction downstream of the TCR, and we'll look at similar concepts downstream of the BCR. See you there.